You are listening to Neurosalience, the OHBM podcast.
Welcome to the OHBM Neurosalience podcast. I'm Peter Bannattini, the host, and on this podcast, we talk to people in the field of neuroimaging, experts, luminaries, rising stars, and discuss their work, issues, and anything else in between. And today, I had the honor of having three guests, two luminaries and one rising star. The luminaries are Tom Nichols and Jack Van Horn, who've been in the field over 20 years. It's really pushing data sharing and data standards, and specifically with fMRI, but not completely limited to fMRI, also including structural MRI and other things. And the rising star is Remy Gao, who's been extremely active through his career, his growing career of sort of trying to change the culture of neuroimaging, of promoting these best practices of reporting all the steps of analysis and sharing data and pooling data and how to best go about that. He actually took basically the group had a paper called COBIDAS, and a movement in some sense called COBIDAS, which stands for Committee on Best Practice in Data Analysis and Sharing. And he made it into sort of an e-COBIDAS, sort of a checklist. And it's still evolving, as we have new analysis techniques and new goals for our data as well. So in this discussion, we talk all about everything about the origin of COBIDAS, and also some issues in, data reproducibility, how to share data, how to report on metrics, and not only that, sort of the philosophy of doing this, and we kind of get into the details and some of the challenges. How do we grapple with this problem? I don't think anyone thinks that will solve the problem, but creating a culture in which everyone's aware of what the best practices are in terms of reporting and even analysis to some degree without being prescriptive is a good thing for the field. And I think it tangibly helps the field. So it's a great discussion. I hope you enjoy it. Thanks. Thanks, all three of you, for being on the Zoom, and looking forward to a good discussion. So I'd just like to start off with just a quick question. People hear the term COBIDAS, and I need more coffee this morning. And people wonder, what motivated it to form, and what was the process by which it actually came out and became real?
Well, I guess I'll start with this particular lead-in, Peter. Thank you so much, by the way, for having us. This is exciting to be here to join you and Tom and Remy. So the start of this idea really grew out of a strategic planning meeting that we had when I think Karen Berman had taken over as the incoming OHBM chair. And we had a strategic planning meeting, which I believe, as I recall, was in Geneva, Switzerland. And we started there thinking about how we wanted to see the organization grow and evolve and mature. At kind of a strategic point, we kind of knew that the 25th anniversary was coming up, and we wanted to make sure that we were taking steps to demonstrate that human neuroimaging in particular was a mature and reliable science. And we recognized that notion of reliability and reproducibility was something that we were concerned about. And we went about asking ourselves, well, what could we do to promote reproducibility, data sharing, sharing of not only data, but also of software and other methods, and how could we do that better? We realized there was this reproducibility crisis, and at the time, neuroimaging was kind of being held up as having a lot of small sample studies that were reporting things that other people could not reproduce. And So I and yourself, I believe at the time, and several others, we put together a little policy statement saying that the OHBM was committed to moving in this direction to try to find ways to encourage reproducibility and the openness, the open science of neuroimaging. And out of this came this notion of creating the COBIDAS committee. And I'll let Tom talk about that in a second. When we were thinking about this, and I was kind of asked to kind of identify somebody from our community who could carry this thing forward and serve as the point person, I immediately thought of Tom Nichols. I have known Tom for many, many years, and if there was anybody who could really be the leader of this, it was going to be somebody like Tom. And I have to say, and I hope you don't mind, Tom, that I could not be more pleased with how you carried this forward in your leadership on this. And it's interesting that we're here now, and hopefully going to see a revision of this coming up. But that's how it got started. Maybe Tom, maybe you can carry it forward from here.
Thanks, Jack. Well, so it was in June 2014 in Hamburg where the council approved a statement.
Okay.
That's right, a statement on neuroimaging research and data integrity. And that basically outlined a number of issues that really needed to be addressed. And the way that the council chose to take it forward was by creating this committee. And I have to congratulate you on creating the name Committee on Best Practice and Data Analysis and Sharing.
It was both Nico Kriegeskorte and myself.
Oh, Nico.
Nico is there.
That's right.
We pulled that one kind of out of the ether somewhere.
It is both pronounceable and leads to good, unique keyword searches on Google.
Yeah.
So yes, you asked me to lead the group on behalf of council, and I solicited volunteers, and I really tried to get a range of people with expertise, but also across mid-level to senior career level, and just set about this process of trying to sort of establish some good practices. And what we realized really early on is that it just is not feasible to just sit down and say, this is the right way to do it, because there are just so many different things that people do. Even we quickly realized, okay, we got to focus this on just MRI. And if we had to do it again, I might even fractionate it even into sort of structural versus functional or other divisions. But at the time we said, okay, everything having to do with MRI. And by and large, it's just there's no way to just say, do it this way, don't do it that way. But what we could say is that we really need to focus on transparent and complete reporting of what people do. And so that became the focus. We identified a range of different sort of domains, a total of 7 of them, everything from experimental design through data sharing and reproducibility. And in each one of these domains, we identified what are the important things that you really need to communicate when you report your results. And then we also produced a table as an appendix, just big, long list of checklist of things that you really should report. Some things which may not always be appropriate, other things that you always really should report on. And that's what happened. We took a team of over about a dozen people, and by 2016, we published that result. And actually, I'm really proud of the fact that we released this to the membership of OHBM, and we solicited input. And I think we got close to 100 different sort of individual comments, and we acted on each of these and responded to them, updated the report, and then finally submitted it to council, and it was approved. And it really was, in that way, a community effort. And then finally in 2017, we published a Nature Neuroscience commentary, just sort of discussing the process and sort of what we learned from that. But really, really glad we're still talking about it, and it does still get referenced, and I think it's an important document. And I'm looking forward to getting the band back together and updating the COBIDAS MRI report.
Yeah. So Remy, do you have anything to add to that, or?
Yeah, I was just wanted to mention, maybe for like listeners who might not be aware, that also after that came the COBIDAS for EEG and MEG that got released more recently. So that's I think that the first report for MRI sort of laid a good groundwork that could then be reused later on by another team of people to sort of carry on the torch, I would say, with other modalities as well.
That's one of the things which I think was important about the initial effort led by Tom. You really kind of set the standard for how we set standards. And I think you made an important point about it's just too difficult to go in with a bunch of thou shalt and thou shalt not kind of idea, because there's so much variation. And really you have to kind of look at an evidence-based kind of dispassionate, what is evidence-based best practice, and go on that. Now you'll have to update that, as we're talking about a little bit here, every once in a while, but that's the best way to do it. And I thought the way that that was done and how others have then built off of that for their other sets of best practices has just been ideal. So, and the publication of releasing it to the community so that you have the full version, and then getting another version that's more the summarized version in some place like Nature Neuroscience. That's fantastic. Right? So it's just an ideal outcome.
Yeah. So the idea then with COBIDAS is that with a transparency of reporting your practices, without there'll be some sort of a awareness and feedback as to what worked or didn't work, or maybe trying to better replicate what was done. It still seems it's interesting because it's a tough problem. It's a really hard problem because as we're learning, I mean, there's so many. Even as I think about this, I mean, there's COBIDAS addressed all these stages of decision making and processing, and so many different things you can do. It seems that that's sort of like, there's layers and layers and layers of decision making, and at least to me, that was kind of brought out even further, in the Nature paper, the first author, Botnick Nezer, basically showing that, even with pretty solid groups doing reasonable stuff, they all analyze, all these groups analyze this one data set, and they got, I don't know, Tom, I think you were on part of that paper, but or maybe not.
Remy too, both of us.
No, Remy, both of you. Sorry, and they got different results. I wonder if there will be another iteration of that paper, sort of dissecting. Maybe, I mean, the idea of the paper was that it's good to have a consensus. It's good to have, comparing different methods, but also it would be good to sort of use that to dissect where things diverged, and maybe use that to guide future COBIDAS and potentially.
Sure, I can say a little bit about that and let Remy say more. What struck me about those results is that, yes, of course, there was such incredible differences, but what was notable is that those differences were by and large not explained by pre-processing choices like software and sort of particular, to use nonlinear versus linear interpolation for the warping. And that the most of the differences seem to be driven by choices made in modeling the fMRI signal in the individual subject, so-called first level model. And that was in choices made by the individual groups and also implicit choices that are made by the software. On subtle things like orthogonalization of predictors and the like.
Yeah.
So that was notable, and that has led to some people saying, well, that's pretty exciting in a way, because it says if you do want to explore this analytical variation, maybe you don't have to build some fancy thing that and master 7 different software packages. Maybe you can just use your one and then try a bunch of different options in terms of the way you model it. But of course, that leads to what they call a multiverse analysis. And then you've got this extra challenge of how do you integrate all those different results. And that was, of course, the main role of the paper. And that's something I'm interested in doing more work on. I think it's a relatively underdeveloped area. It's like meta-analysis, but in meta-analysis, you have independent studies you're trying to combine together. And in megaverse analysis, it's all the same data.
Right.
Then you just analyze it with a bunch of different ways. So, Remy, if you want to add. Okay,
yeah,
go ahead.
Remy, yeah.
Oh yeah, I mean, I might just make a side comment on what you referred to as like what people call like the NARP study, because that's what I think now it's becoming known as. One of the things first is that we learned that in PET neuroimaging they had done something similar a few years back as well. And I think that now there's a current project to do the same with EEG, I think. So that's also interesting to see what's going to come out of that one. But one thing that actually links the NARPS project and study with COBIDAS is actually all the teams were asked to report their methods, their analysis using the COBIDAS standard, right? So that was like really essential to make sure that we had a standardized way so that the results of the different teams and what they had done could actually be compared. So I think that's even just there, that already shows the usefulness of COBIDAS and of like having standardized reporting, a way of reporting the methods.
And that's really interesting how, right, you sort of discovered these other subtle aspects of modeling the hemodynamic response and things like that drive a lot of the, that seems counterintuitive to some sense, I mean, or maybe not really completely what people think of first when they think of sources of variance, but yeah, no, and so then the hope then is that the consensus is sort of the truth in that regard, as opposed to, finding, right. There might be one best way to do it, and then so
I think unfortunately, I think we'll need many more of these megaverse analyses before we might be able to identify modal or sort of best. I mean, unfortunately, a lot of these things relate to things that people could just reasonable people could disagree on. I mean, I'll say one thing of hope, and that is that there are a lot of this study was a mixed gamble study, and it required some quite subtle modeling. So basically, some of the questions had to be answered with a parametric modulation of one regressor with another, and those things are quite subtle and can be, I think that's part of the reason why we saw such differences. If this was a block design, with one main effect, I think you would have seen a lot less variability. So I mean, I think that's telling you something about fMRI. It's not that there's, this was a good stress test, and there are a lot of people that do studies that need this level of complexity. But just to say, not all studies involve this level of complexity in their first level modeling.
Yeah. Also, at least what I got from the paper as well is it sort of speaks to a little bit on how people draw conclusions. I mean, of course, you always have to make a map and you have to threshold, but then you could draw different conclusions based on, I mean, at least I think, as I understood this paper, is that they looked at the spatial correspondence between the threshold results to some degree. And that's it feels like it's very sensitive to, what threshold you choose. And, if you sort of had an unthreshold map and looked at, it seems like results might converge a little bit depending, based on the type of post-processing or interpretation you make of the data.
Yeah, it does worry me sometimes that the most important sort of system in a brain mapping study is the visual system of the scientist who studies the thresholded maps.
Yes.
And that sometimes might not be avoidable, but I'm always keen and energized when I see studies that come up with ways of sort of casting the problem in another way, or either bringing it down to individual questions. And I should say the NARP study did demand that each team produce yes/no results for very focused hypotheses. Right. And I think that would be, if we all were sort of challenged to do that more, I think that'd be good. I mean, there's a lot of complexities in the data, but in the end, that's what people are going to take away from the studies.
Yeah. Okay. So talking about, future COBIDAS efforts, what did you see were potentially, how has the field evolved maybe in those few years, and in what might be the gaps in the current COBIDAS that might need to be filled with regard to reporting?
Sure. Well, I'll start at a high level, then maybe Remy has some other comments, and that is, so there's some things that I think, anytime you look at something again, you might, even if we'd done it just a year later, we might make slightly different choices, and I think that's something that we'll do, revise, and probably make it slightly better that way. But at another level, there is the things that, MRI is exciting because they're always coming up with new ways to acquire data. So for example, I believe multi-slice or multi-band imaging wasn't really much of a thing when we first did that report, and now that is. And so there are new technologies that are evolving that it will be important to integrate those into the report. And then I think there are just kind of the cultural changes. So I think we were very cautious about how hard we pushed about the importance of data sharing and publishing your data. And I think we just have a greater level of confidence to say, yes, this is really important and it really needs to be done. So I think that's those three things, just sort of minor sort of revisions, adapting to changes in the field and in terms of technology, and then adapting to sort of the state of open science as it is.
Yeah, I've been working with people at the NIH, and there's one scientist who has a very subtle study and collected 150 subjects and looking for a very subtle effect, and he found like extremely low interclass correlation values, and he didn't know why. And it was a very much of a learning experience as I worked with the team with Bob Cox and Daphne, we're still working with trying to dissect why. And it even comes down to things like, obviously the time series might vary. They initially were saying, oh, it's the problem with the scanner. and it wasn't a problem with the scanner. And then it comes down to sort of even subtle things like looking at each individual, how you pick your regions of interest, and having a little bit of warping where you don't expect it changes things tremendously. And he was all ready to write a paper saying fMRI is not useful. I'm like, no. But I do think that that it's not useful in this particular case or something like that. But I think it's important to try to head those off. I mean, there's always things like that come up, and it does seem like there's even subtle things like that your average practitioner will miss. And one of the things that I also saw from that first paper is that your multi-study paper is that one of the fallouts was like, oh, well, maybe if we have as much automated as possible, that would help the reproducibility. But at the same time, it seems that in this particular case, there's an art to, you really have to look carefully at each individual subject's data and every step of the way, and an expert needs to tweak it. But at the same time, if you're not an expert, you could be making it worse. So it's a tricky balance in that regard too. So
automating things works to a point. It can be very helpful, especially when you have a lot of subjects to kind of grind through. But I just don't think you can take the human completely out of the loop. You should always be checking. I could, numerous stories that I've experienced where somebody entered the wrong number as part of their pre-processing script, and it caused all the brains to shrink to half the size they should have been. And then when they got fit to the template atlas, they didn't fill up the atlas. And so when they did their analysis, they just got basal ganglia activation because all the brains were that size, right? And it was just cookie cuttered out the basal ganglia. And when you went in and found that, and then redid it, you found that, okay, it fleshed things out and then you got cortical activation again. But it's easy to do. Somebody misses a decimal point, they don't catch it, and then you've run it through a script, and then that's it. It's always important to kind of double check and triple check what you've done just to make sure, so you don't make stupid or careless errors.
My favorite was someone was combining different contrasts from different studies, but it was the same sort of gambling task. But in one, the contrasts were defined relative to pence, or I think it's probably euro cents. And the other one, they're defined in euros. And that actually was enough to create numerical underflow problems that was so different. It just had really bizarre errors until we figured that out.
There's some strange things that can happen if you're not careful like that.
I don't know, have a good answer really for your question, Peter, because it really goes beyond sort of reporting standards and just saying that there's a lot of training that's needed to do MRI well and fMRI.
You're right. I mean, it really is. I mean, fMRI is really robust, and for the most part things work. But right, when you're starting to push the limits, a certain amount of skill and trading, it becomes much more important.
Let me just loop back, because I was thinking Remy was going to jump in, but you're talking about sort of things that are to come with COBIDAS. I think something that's really important that Remy's been working on is actually making COBIDAS more user-friendly. So if you look through the COBIDAS report, there are literally, 40, 50 pages of these tables of did you do this, did you do that, And it's really overwhelming, and a lot of it doesn't necessarily apply to your study. But maybe, Remy, you can talk about the effort you've been involved in with eCOBIDAS. Make this easier to use.
Yeah, that's definitely, I think, that's definitely one thing that I've been very passionate about, and I've been able to find people who are really passionate about this, which is, as you were saying, the COBIDAS report is fantastic. It's a great resource, but going through it is 50 pages, even just the table. I mean, maybe not 50, 30 pages of just tables at the end, which is great, except that I think there are two issues with that, is that many of the items that might not concern you, like there are 2 pages on arterial spin labeling. If you don't do that, you might have to flip through them all the time. And what we started doing with this was to sort of, first of all, just turn that into an actual single table, and that now goes up to 400 items. And that is just completely overwhelming for anyone to actually go through. And we're trying to transfer that into an app where you would only be exposed to the items that are relevant to your use case, right? So you don't need to sort of fill in 500 items or to flip through entire pages of things that do not concern you, because I think one aspect that is really important for a project like this is that adoption by the community is really important. If you create those fantastic guidelines, but no one uses them, it's, you've just been wasting your time pretty much. So, adoption will only work if the usability is there as well, right? Otherwise, it's going to be restricted to a few people who just are really passionate about reporting their methods really accurately and will take the time to do it. But if you can't manage to make it easier for every researcher to do that and to quickly show them that there's an advantage of doing that as well, then I think it's going to be hard to sell.
Yeah,
so
well,
that's sort of the effort we're working on now.
Yeah, and that kind of speaks to the whole culture. Where are the incentives to actually do this? It's partially to have your data accepted, and everyone wants that, and everyone wants it shared. I mean, I guess some people don't want it shared. Some people,
There's still a few out there who don't. Yeah.
Yeah. And so obviously you want to make it as easy as possible to sort of minimize that amount of work that goes into it. But then are there, like, what would you tell people the incentives other than just being a good thing? It's kind of like, recycling or things like that. I mean, people do it and they get in the habit of doing it. Is that what we're relying on here? Or is there some other way of leveraging?
The goal, sort of like one of the end goals, sort of pie in the sky type of like things that I'd like to get to is when people have clicked through a whole checklist to describe what they've done, you'd actually be able to automatically generate sort of part of the method section of their study, right? So people would not have to do that because it would be done for them in a sort of system, automatically, right? So that would be, and the advantage of a checklist, you make sure you don't forget anything, and then people sort of, that's the carrot they get, is that here's your method section. You have some tools that already do that. So we were talking about pre-processing. So fMRI prep, which is one of those pipelines that exists, will generate the method section corresponding to what pre-processing has been done. So the idea is a bit the same, but we're trying to make it more flexible. But that's an end goal. We're not there yet, but that's definitely where what we're aiming for.
Okay.
It's kind of interesting about trying to convince people who've been doing fMRI for a while that they might need to do something else or need to report more. One thing I've tried to do is incorporate COBIDAS into the teaching I do of both undergraduates and graduate students. And so, like, I ask them as they're looking at papers from the literature, are they compliant with reporting standards like COBIDAS, for example? And a lot of students, it's kind of interesting the reaction that you get from them. They aren't aware of it, of the COBIDAS report. And when they look at it, usually the reaction is, well, this seems to make a lot of sense. And so, I'm encouraged by that some people who are entering the field or maybe entering the field will already have this as kind of a baseline, right? This will be the norm for them. And so hopefully as new generations come into the field, that they're already starting off at a good place.
And I'm just going to jump in also say another really good pedagogical aspect of COBIDAS that I have found is that when I've had people helping me on the project, sometimes they realize they don't even know what some of the questions are, what they mean, right? And therefore there's a really good potential to sort of educate early career researchers to why you should report this and why it's important and how it can affect your data. There's I think a good sort of education aspect to this project as well.
Yeah, it definitely does seem that you're right. It will iterate and people will get educated on, okay, so I'm reporting this and I need to be aware of this and raise the standard of the research everywhere in that regard. And I think, I think it's wonderful to try to make it, a sort of an easy checklist as much as possible. So maybe I'll shift towards a little bit more towards the goal. I mean, obviously the goal is to make the data, to share it, to make it more reproducible, to fend off, criticisms of how fMRI is useless or whatever. And ultimately it's to use fMRI clinically. I mean, and it seems that at least one aspect of clinical use is to generate biomarkers. And you're looking for like, vanishingly small effect sizes, and you need really clean data. Some people argue, though, is this really necessary to pool such large data sets? Why not? Some people still stick to their N of 20 and expect to get, they get results and they write up a paper and they feel that's good enough. Some people do one subject or 5 subjects, hundreds of times. That might be good. I'm kind of curious what your thoughts. I mean, obviously, COBIDAS sort of ties into all of these, but especially though for those large consortiums that have many different scanners that want to pool their data to maybe generate, something like the UK. Well, even the UK Biobank is unique because that has the same scanner, and it's just the same scanner spread around to some degree. So there's some control there, but there's some consortiums like ABCD that uses different vendors. But there's also wide open data sets where you just simply take whatever you can get. Are some more worth doing than others? Or is there something to say for these N of 20 studies that produce something? I mean,
I'll take a stab. And I think it as you're hinting, it depends on the goal. I would like to say that I think there are some effects out there that are incredibly robust, and you probably don't need much more than 20 subjects. If you're looking for the main effect of working memory in a N-back continuous performance task, that probably does it. You get huge responses in response to that task. Now, instead, if you're looking for subtle modulations of that task, correlated with some intrinsic trait of individuals, you may be struggling with 20 subjects to tease that apart. So I think, for main effects or sort of just asking primary questions about different tasks, it's possible that with small sample sizes, you may be able to suffice. That may suffice. But it seems like most of those things are kind of been sorted out. They got their start in PET even before they're then replicated and further explored in fMRI. And we're to a point where now we're interested in these subtle things. And, how does brain responses relate to subtle health traits or socioeconomic status? And I do think that for those sort of questions, I think we're getting to the point like the geneticists have gotten to, that you really need large data sets if you want results that are going to generalize. And things that boil down to correlations, basically you've got a noisy measure of brain activity and a noisy measure of some trait of the individual. You just need really large and hopefully as diverse samples as possible to get results that are going to generalize. But the converse is that if you're interested in elucidating, the subtle structure of a behavioral effect, then it may actually be better to then take a small number of subjects and really scan them a long time to really kind of get rid of measurement noise and really understand. And of course, that's almost taking the lead from non-human primate work, where you do just have a very small number where but you collect a lot of data on those small number of individuals, and you can see how that could work for some specific questions about the structure of cognitive processes.
Yeah, Remy.
Yeah, I just wanted to jump in to say, well, I mean, first, I think it's still good to remember that in some fields, it is really hard to get large sample size, right? I mean, there's something I sort of discovered in the lab I joined here, where they do a lot of research with blind participants. It is really hard to get a very large sample size when you work with blind participants. So in this case, I think the focus is definitely trying to sort of making sure that you get data of really good quality, and make sure, I mean, once again, I'm going to go back to it, but make sure that you report your methods accurately, so that if people want to do meta-analysis, then they've got all the information about your sample, your design, your acquisition parameters, so that they can combine information across the in an appropriate manner. Right? So I think that's another way of how we think, making sure that you've got standardized guidelines for reporting can really help as well, even for the small sample size stuff.
Yeah.
And even these big studies, UK Biobank it's amazing amount of data. Human Connectome in the US, Human Connect, ABCD, some of these other ones, they're really amazing resources. But even if they all decide to use the same make of scanner, they're going to use a Siemens 3T Prisma, for example, still site-to-site variation exists. you want to be a little careful about just combining all that without taking into account the acquisition sites, because even like, successive serial numbers off the assembly line probably are going to produce slightly different, scanner specific effects that you want to be aware of.
Yeah. There's a workshop at the NIH September, and it's interesting. Hugh Garavan was talking, and they're discussing the problem with some of the ABDC preliminary results showing not much of an effect size, and then they thought, well, maybe it's the scanner differences and whatever, and it might be, and certainly that would be helped with more data. But also, I think talking about more of the subject parameters, more of the behavioral measures, more of their demographics. And what Hugh did is he sort of parsed the data based on the demographics, and he started getting much larger effect sizes. And obviously, it's a little bit tricky. You don't want to, select your data based on, getting results. But if you have an objective way of parsing even variability in, we're all different. And and there might be some subcategories of people that fall into a response, pattern of activation or even timing or whatever, that might be very different. And sort of trying to he sort of said, well, let the data sort of show me what's different, and then I'll see if that corresponds to differences. And so once again, it comes with clear reporting and clear metrics all the way from acquisition to, subtle subject demographics even. Once again, depending on the study. That may or may not be necessary. So,
well, I think these are the sort of questions that we can finally look at when you have the 4 and 5 digit ends, to be able to discover what is the heterogeneity in your sample that you just really don't have the luxury of doing with smaller sample sizes.
Yeah, I completely agree. I completely agree with that. You sort of answered the questions, like all of these types of studies, the small n, large n, different vendors, same vendor are worth doing. And obviously we try to, as hard as we can, to keep the variability as low as possible, but reporting sort of fixes or helps to fix that problem just by simply allowing you to sort of parse the data in some way afterwards. It would be great to sort of try to figure out ways of algorithms for normalizing the data instead of parsing it. Potentially.
I agree. The first step is getting that all recorded. And in the ideal setting we would be in is that every paper would have a machine readable appendix, or file that would describe, basically the answers to the COBIDAS questionnaire that would begin to give some data that would then help us parse apart results. So that would be in the role of doing meta-analysis. You could then start trying to controlling for subtle differences in the way studies were carried out.
Exactly. Yeah. And those small end studies, being respectful of what's in the COBIDAS, it's a measure of good faith that you've done your science and you're reporting it as fully as possible, so that those kinds of that filtering and accommodating these subtle differences can be accommodated for, so that we can try and clear up where some of the source of this variation comes from.
Yeah. So also related to reporting, I guess, there might be, we even thought of there some discussions of reporting time series metrics. And that's, to report that well, it takes a certain amount of software to pull that information out. And there might be, differences in the software. Is it, where do you go in terms of, do you say, well, these are the metrics you want to report, but then let's say everyone uses different software. Russ Poldrack has some, MRQC sort of software, and there's other groups that have software for doing this, and it's all different. And they'll think they're reporting the same thing, but it could be a different thing. So do you think there should be some sort of code standardization as well in terms of software for gleaning that data to report?
I think standards for basic QC things, I think would be great for interchange, because I think, I'm sure you're aware of the work that has been possible with Russ's fMRI QC. If you opt in, you, or maybe failed opt-out, I'm not sure which way it is, summary data is collected on the studies you run through fMRI QC, and that's just an amazing resource to see the heterogeneity of the data and the differences. I had explained that differences in quality. So I think that would be an important thing. Maybe COBIDAS can contribute to that.
And yeah, if people can agree on what the equations are, at least, then how they get implemented is kind of another thing. But because different people want to use MATLAB, some people want to use Python, some people like ANSI standard C. But however it got implemented, agreeing upon what the equations were that describe, say, image quality, those hopefully could be a little more easily agreed upon.
Yeah, I think we just have to be careful of getting to a point where it is tempting to say, "Oh, come on, we all agree on this, so let's all use this one software."
We've seen that happen. Yeah,
we've seen that. Well, in some ways, the cluster failure was about that. Everyone thought like, "Oh, random field theory. That's all. Yeah, everyone trusts that. That's good. It's all being implemented the same way." But basically, it's science. We always have to keep on going back and checking these things.
Well, and we don't want to crush innovation either, right? We want people to kind of like be pushing the edge of the envelope, as it were, and trying new things. We don't want to stop that, but it's nice to make sure that people are using, they calculate the mean in the same way.
Yeah, even something like that. So it's tricky, and it can make a difference. But yeah. Oh, Remy, you were going to say something?
No, I guess I would say that the bare minimum in that case would be just at least tell us what is sort of the pipeline you use to do your QC and what version you use, right? That would be like, at least we know, and we hope that by this we can point to the equation that was used behind it. That would be like the minimum, at least, I would say.
Yeah. No, I see. In some regards, I see certain software obviously converging, but at the same time, I mean, it's a little scary. I mean, when as we get we go to high resolution, different vascular calibration approaches, different parcellation methods, deep learning. That's a black box in itself. I mean, how do you think COBIDAS could potentially, address these things in a, I don't even know if it can or not. I don't know whether there's.
Yeah, I mean, when we discussed this in the committee originally, it's everything is it's research, right? So it's a moving target, and also I was involved in another project called the NIDM, Neuroimaging Data Model, and this was trying to say we are going to develop a controlled vocabulary that will describe every facet of a study. Well, we quickly realized that's, for example, in resting state fMRI, there are just so many different ways that people do it. Some people use regions of interest, some people use independent components analysis, and there was, relatively few, there are some pipelines out there, but a lot of people, just kind of roll their own and do lots of the details in different ways. And we were at a complete loss of how to do that. And so we took a problem that was more manageable. We focused just on task fMRI, where, for better or for worse, there were just a small number of packages that everyone used, and basically those packages were doing roughly the same thing. But that exercise showed me how challenging it is to really thoroughly and completely describe what people are doing, even in task fMRI. My favorite thing was we had a 1-hour discussion of what is a threshold. And is a threshold that's specified as 0.01 as a probability, is that the same thing as a threshold that was specified as 2.3? And should those be represented semantically as different things? And it just shows you that if you are really going to capture these things precisely, you need to get to that level. And that was the decision we made with COBIDAS, to say, listen, this is just, there's too much in here. We are just going to try to say, these are the things you need to report.
Yeah.
In all fairness, it's a little unfair, I guess, to Tom and to the folks involved in COBIDAS 2.0 to overload them too greatly.
Right.
Solve everything, Tom.
Yeah, no, it's not solve everything.
And this is one of the reasons why OHBM has come up with its best practice committee, is to help to encourage Tom to take COBIDAS and Remy to take COBIDAS and bring it up to the current standard. But at the same time, look at other domains, like, for example, the quality control issue. That it's related, but it's maybe at the other end of the processing pipeline where some more attention needs to be given. And then there's other modalities too. It's not just fMRI, it's EEG and MEG, as we've seen. There's an effort going on to look at how we describe and report on network analytics. And so we're looking forward to that. And there will be others. And so it won't all be COBIDAS. There will be several of these. And that's why OHBM decided to move in this direction, because there's just a lot of different dimensions to what we think of as making neuroimaging science reproducible.
No, this is, I mean, it's a hugely daunting multivariate challenge. And yeah, I mean, and actually the fact is it's the alternative of not doing anything is certainly that not desirable at all. I mean, we have to do something. It's important to keep and actually try to, keep changing the culture of neuroimaging such that everyone's aware of sources of variability and wanting to, think in the long term in terms of sharing and having results that are actually reproducible. I guess we're getting close to the end of the time. There's obviously so much to talk about, but so, I mean, obviously, is there anything else you want to add as far as, there's OHBM making the best practices sort of efforts, there's COBIDAS, and are there other groups? I think it's worth mentioning if there's other groups that we can work with as well that are doing.
Definitely. The International Neuroinformatics Coordinating Facility, the INCF, has really taken on looking at standards in neuroscience and neuroinformatics generally. And they're a very interesting ally in this effort. Many of us who are involved in the OHBM effort also have participated in INCF efforts. And there are a number of other organizations as well who are following what we do in this space, and they want to learn from us too. So we have an opportunity to partner with other international leaders in this space and to influence other efforts around sciences that utilize neuroimaging methodologies. Remy, I know, is deeply involved in several other efforts. Remy?
Yeah, no, that's true. I mean, trying to sort of coordinate with INCF would be sort of essential. I agree.
Yes.
That's for sure. I'm actually just going to point at another group I think that or sort of community is all the people working on BIDS, which is the brain imaging data structure, because a lot of the effort around standardization at the acquisition stage is definitely something that that is at the heart of the BIDS project, I would say. And if we can avoid reinventing the wheel on the COBIDAS side by reusing some things that already exist over there, I think this is something that should clearly happen. So we're trying to sort of connect the dots on that front as well. So also because there are other modalities that are already part of BIDS, and if COBIDAS wants to sort of like have an eye on that later on, then it makes sense to sort of like keep those different moving parts connected somehow.
Yeah.
Yeah, no, I think echo that BIDS is essential because, one of the recommendations from COBIDAS is to share your data and share as much of it as possible. And BIDS makes it gives you a way to do that, and basically tells you, here's a way, if you follow this outline, this approach to sharing data, you're going to make your data maximally useful to other individuals. And in fact, having all this data in BIDS format has then sort of supported this ecosystem of BIDS apps or tools that take as a starting point the data that will exist in BIDS format. And it's just been amazing to see that grow. I guess the only other thing I'd add is that I think for people who are actually in a lab or, just in a small group wondering what they can do, and it really seems daunting. And all I'd say is to, every little bit helps. So, even if you just pick, one part of the tables to really pay attention to, or, doing incremental things is much better than doing nothing. And so I think everyone should have that in mind, that everyone wants to have that perfect paper, that everything is perfectly reproducible and everything's in notebooks and everyone could rerun it. And but that takes a lot of effort, and that's where we want to be. But every little bit you can do to maximize the transparency, share as much stuff. Maybe your code isn't perfectly documented, but sharing that code, sharing the text files that gave rise to your experimental design, all that is supporting the sort of the reusability and sort of the transparency that will help make your data as useful as possible to everyone going forward.
Yeah, well, thank you. Yeah, definitely. I mean, sharing helps not only spread it, but gives feedback to help improve it. And so, Yeah. All right. Well, I mean, certainly what you're doing, what all of you are doing is critically important to the field. And it's a natural aspect of how the field is evolving, should evolve. So, yeah, and it's not a clean, solvable problem. It's just one that we grapple with, and by the nature of grapple with it, we keep on improving things. So, all right. Well, thank you very much and this is a really, really, at least in my mind, informative and helpful podcast, and definitely there'll be a lot of work to do in the future as far as this is concerned. So
Thank you so much, Peter. This was
Great.
really
Thanks,
great.
Peter.
all right.
Thanks, Peter.
Thank you.